Thanks, everyone. I'm delighted to be here. Here's a bit more epidemiology for you right at the end, looking at the microbiome of built environment and health effects. All right. Okay, so I have this, let's see where I should point here. This schematic diagram up here that we've all probably had in our minds, or at least some of us have thought about, which is that building characteristics and human behaviors can influence the microbiome of the built environment, and we believe that they do. There's a bidirectionality between the microbiome of the built environment and the human microbiome. They can each affect each other. The microbiome of the built environment has effects on human health outcomes, for sure. I'll show you evidence of that in this talk. And it may directly influence human health, or it could potentially influence human health by alterations in the human microbiome. So I'm going to go through this bit by bit and give you a little bit of pieces of evidence for each area in the diagram. Okay, so first of all, what's been looked at in terms of the microbiome of the built environment and its influence on human health? So let's start with the exposure biomarkers here. So there have been multiple microbial exposure biomarkers that have been looked at in the indoors, in homes in particular. These include endotoxin and 3-hydroxy fatty acids from the lipid A portion of LPS from gram-negative bacteria. Muramic acid, which is mainly a marker of gram-positive bacteria. And then fungal biomarkers like ergosterol, this is like fungal cell cholesterol, and 1,3-beta-D-glucans. We also have culture data that we can use to assess exposure. And then, of course, we're really here to talk quite a lot about microbiome sequencing characterizations like 16S sequencing for bacteria, fungal ITS sequencing, and shotgun metagenomics. Now, on the health side, I tend to think of studies as being grouped as environmental health or occupational health in response to these microbes. So a fair amount has been done, and Susan Lynch spoke today already on infant and childhood health, WHEEZE and asthma outcomes, allergen sensitization, and immune outcomes, adult health outcomes like asthma severity, and then occupational health outcomes, including airway obstruction, and even some studies on cancer as well. Okay, so the first bit of the diagram here, I'll go over some of the work that we've done looking at building characteristics in human behaviors and the way that they can influence or how they're associated with the microbiome of the built environment. Okay, so here's some work that I published in EHP a while ago that showed that pet ownership, home dampness, cleaning, and race are associated with home microbial biomarkers, okay? And so here I have the percent change that we observed, and the microbial biomarkers are down here along the x-axis. So fungal glucans, gram-negative 3-hydroxy fatty acids, endotoxin as measured by the RFC assay, and muramic acid all show a percent increase with dog ownership. Okay. Now, one thing I should mention, I made these plots, but what I don't have up here is the R-squared, or the percent variability in these markers that's explained when we ran our models. And that generally is quite low, okay, on the order of 5 to 15%. But we do see significant relationships with some of these characteristics. Okay. So here we have water damage. Fungal biomarkers like ergosterol and glucan are higher when participants report water damage in the home or visible mold or mildew. Gram-negative biomarkers are higher, and also muramic acid is increased as well. And these are data, I should mention, from our home allergens and asthma cohort study that was done in Boston. Okay. So now a few more home characteristics here. Cleaning frequency is associated with lower microbial biomarkers, including fungal ergosterol, gram-negative 3-hydroxy fatty acids, endotoxin by RFC, and gram-positive biomarkers. And interestingly, race is associated with lower levels of gram-negative bacteria as measured by RFC endotoxin. And this association was present in multiple regression models after adjusting for all covariates. And we don't really know why we see this, but it is kind of an interesting result. Okay. So that was a bit about home characteristics and predictors of some of these broad biomarkers from our home allergens and asthma epi study. Now I'm going to show you some data from a pilot study that we did, okay? And the title of this slide is Xenobiotic Degradation of Household Chemicals May Be Overrepresented in Indoor Bacteria. So I should mention that the data I just showed you was done, the samples were from about 300 homes, okay? So we took a subset of the house dust samples that we'd analyzed previously, about 12, from the Boston area birth cohort, so house dust samples. Then we also had some outdoor air samples taken from a different study that was done in Toronto. And we sent all of our samples to the Jackson laboratory that does our sequencing for us, okay? And they ran 16S sequencing for bacteria. They did ITS for fungi as well. But they took the 16S sequencing data and they imputed the functional metagenome of the samples using this tool called PyCrust, which is available from Curtis Huttenhauer's lab at Harvard. Online, you can do this. And so they imputed the functional metagenome of the bacteria, the functional genetic potential of these microbes, okay? And they handed us back the data. And it was kind of interesting. They put both sample types on the same graph. We hadn't initially asked for that comparison. But what was kind of cool was that in the house dust samples, which are in blue, we saw an overrepresentation of the potential to degrade caprolactam, limonene and pinene, and geraniol. So caprolactam is a nylon precursor, okay? Limonene and pinene and geraniol are additives to household cleaning products. So it was sort of cool that these indoor microbes seem to have more of a potential for metabolizing these types of chemicals. And it gives us an indication of the types of chemicals we may want to measure potentially if we're going to assess chemical exposures in house dust. I should also note that the imputed functional pathways in the house dust 16S microbes included degradation of pesticides, bisphenol A, dioxin, and polycyclic aromatic hydrocarbons as well. Okay. Now, other investigators have looked at differences in bacterial and fungal communities as observed by home characteristic. So this is a publication from Donna Miller et al. In 2015 that looked at house dust samples from 198 asthmatic children's homes. And they compared community composition differences for AC use and occupancy, including people and pets. And in the homes that had pets, they observed increased Bacteroides, Firmicutes, gamma proteobacteria, and fusobacteria. There were a number of OTUs within those groupings that were quite different for pets. With AC use, they noticed increased fungal genus Candida and increased Saccharomycetes. There's the Lactobacillus johnsoni finding that they tested out with pets. They found that humans, I believe, had higher levels of Lactobacillus johnsoni, and there was a trend for increased Lactobacillus johnsoni with dogs as well. Okay. So moving on to this other part of the diagram, looking at the microbiome of the built environment and human health. Okay, so I'll discuss some of the work that we've done, our group and others have done, looking at some of these broad microbial biomarkers in the home and culture data and environmental health outcomes, mainly focused on infant and child health. Okay, so again, this work was done in the Boston area birth cohort, the Epidemiology of Home Allergens and Asthma study, and the PIs of the study, or PI is Diane Gold, and Don Milton actually contributed a lot to this work as well, too. I'll start on the left-hand side of the slide here that describes early life exposure to home endotoxin. And in our study, and in others, we've observed that higher levels of home endotoxin are associated with increased risk of wheeze in infancy. But the same elevated endotoxin exposure in early life is associated with decreased risk of allergen sensitization at school age, and I show the relative risks here. And these were findings observed in an urban, suburban type environment that kind of mirror the farm findings, although the microbiota that may be driving these protective effects could be quite different in the different environments. We don't know for sure. And then here's a graph showing longitudinal exposure. So it's clear that the infant window matters, but later on might matter too in terms of exposure. So here we see a decreasing trend for recurrent wheeze that's lowest. So you appear to be most protected if you were exposed to higher levels of endotoxin, both in infancy and at school age. So this is a recurrent wheeze outcome at age 7. Current asthma findings were quite similar to this. Okay, so here's a bit about measurement error correction techniques. So, and this is focused on endotoxin, but measurement error correction techniques may enhance our ability to detect the effects of microbes. So I mentioned in that earlier slide that home endotoxin in infancy is associated with an increased risk of wheeze. And we think that this is probably an airway irritant effect that's going on as a result of inhalation exposure. And so Nora Horick and investigators applied a measurement error correction model that was originally developed by Bernie Rosner that used information from a validation study that paired dust and air samples, okay, to estimate relevant airborne exposure. So in about 80 some odd homes, we had both dust and air samples. We used this measurement error correction model to estimate a surrogate of airborne exposure for everyone in the cohort, about 400 some odd kids. And you can see, here's the uncorrected estimate, really the one I just mentioned to you, the 1.33 increased relative risk with endotoxin for any wheeze in early life. Here's that effect estimate adjusted for other covariates. And here's the corrected model, okay, approximating airborne endotoxin exposure instead of looking at it in just the dust. And you can see that the effect estimates are a lot higher and are bumped up even more when adjusted for ownership of dog in the home and dampness and so on. Okay, now switching gears a little bit to fungi and culture data. And this is work done by Behrooz Beibad. So culturable taxa in the home are associated with WHEEZE, but represent only a fraction of total environmental fungi. So I'm sure that surprises nobody here. But anyway, here's a table looking at the culturable fungal taxa that we found that to be important with respect to any wheeze in the first year of life. A lot of these relationships carry through to later wheeze and asthma. So Alternaria, Cladosporium, and Penicillium were all associated with increased wheeze risk. But our goal is to eventually sequence all of these dust samples to identify other potentially allergenic fungi that might matter. And I should mention too, what's really kind of interesting about this study was that we found that total yeasts were protective for any wheeze, and this effect carried through longitudinally as well. But we don't really know what yeast may be driving these effects in the sample. So these were identified as yeast by the mycologist on the study, Mike Muhlenberg. So when we did sequencing in that little pilot I mentioned to you of the home allergens and asthma dust in about 12 samples, over 40 fungal genera had a possible yeast form. So it would be great to investigate and get funding to do all of the samples to see which yeast might be driving these effects. Okay, so now a note about structure and function of microbial components. Structure and function of biologically active constituents of the microbiome, I'm sure matters for health. And I have a little bit of data here showing that 3-hydroxy fatty acid profiles that come from the lipid A portion of endotoxin give different associations with current asthma when you look at the different groupings. So here's the lipid A portion of the molecule. There can be different fatty acid chain lengths of lipid A, and these are related to the potency and activity of endotoxin, and they vary by taxonomy for sure. And we really haven't characterized very many lipid A molecules, mostly pathogenic ones. But anyway, so here is the findings from our home allergens and asthma study that show that the sum total, okay, of the mid-chain fatty acids detected being greater than the median is associated with protective odds for current asthma, okay? But these other forms of LPS, the long-chain fatty acids, don't seem to have much relationship with current asthma. Some of these in part may be derived from actinomycetes, but this is just to give you an idea of the function and the structure of microbial components is likely key, and we should think about investigating that taxa by taxa if possible. Okay, and so here is the third bit of the diagram, the relationship between the microbiome of the built environment and the human microbiome, and I'll show you some of the results we have out of our Channing Lab cohorts. So to switch gears, I've talked a lot about this Boston area birth cohort study where we gathered the house dust and measured the biomarkers and so on. This is a different study that's been done at the Channing Lab. So the VDART cohort, I don't know if anyone's familiar here with the VDART clinical trial, but this is a clinical trial to assess vitamin D supplementation in pregnant women and asthma incidence in offspring. Okay, and there's an ancillary study that's been done within VDART to look at the infant gut microbiome and asthma. All right. So here are some findings. We haven't quite gotten to looking at the infant gut microbiome and asthma yet, but we will. But initially, we looked at predictors of the infant gut microbiome in these kits. Okay, and about 325 of them. And we see relationships between race, and this is a Shannon diversity index in a multiple regression model, okay? We see associations between race and Shannon diversity of the infant gut, C-section delivery, and breastfeeding. Now, initially when I ran the univariate models or looked at the predictors one by one, we did see some associations for cat and dog ownership. But when I entered all the variables into a multiple regression model, these findings kind of went away, and it turns out that they're really confounded by race, like different racial groupings have different prevalences of pet ownership. Okay. And so what else should I tell you here? We also looked at taxonomic profiles of the infant gut in VDART, and we didn't really see any association between home characteristics and genus level taxa when we ran Maslin, which is a multivariate analysis technique that assesses all taxa simultaneously. Now, is this the last word on these relationships? No, not at all. And I should mention that this study wasn't really designed to look at the environment. It was more about the infant gut, but I figured that I would sort of put this up here and show our findings and say there's not a lot of certainty about them at all. But here is what we've observed, at least in our VDART study. And here's the model R-squared for all these characteristics, 22%. So actually fairly high. Okay, and then here's another study. This comes from the child study in Canada that looked at dust and stool communities and found that, of course, they look very different. So let's see, the circles are dust, the triangles are stool, and there isn't a real clear clustering by pet ownership, yes or no. So these investigators did notice some taxonomic overlap. About 14 OTUs out of the hundreds that they detected overall overlapped in these 20 paired house dust and infant stool samples. But it's a little bit hard to know if it's stool to dust or dust to stool, like which directionality the relationship really has. And just because there isn't tremendous overlap doesn't mean that the overlap that does exist isn't important. It quite certainly could be. Okay, so here's just a listing of analysis methods, some of which we've been using to look at microbiome comparisons. And so, for instance, I used Maslin to look at predictors of the infant gut microbiome, but that technique could also be used to look at built environment characteristics and all taxa simultaneously as an outcome, adjusting for correlations between taxa and so on. LEFSA is another linear discriminant analysis technique that could be used to look at the microbiome of the built environment and how it relates to health. This picks out features that discriminate between phenotypes. So Don actually showed a LEFSA output in one of his slides. Let's see, Permanova is another one that lots of people have been using. And then it's possible to go beyond RNA gene sequencing. Even if you just have the 16S data, there's more to be learned besides taxonomy from it. So I've mentioned pie crusts already. You can impute the functional metagenome using your 16S data. And then there's another technique called oligotyping that people may have heard of. It's a technique used to uncover decomposed, concealed diversity in OTUs. So when we group things by 97% similarity, there's some additional diversity that sort of isn't quite accounted for. And some of these oligotypes may relate to functionally different microbial taxa. So I'm actually mentoring someone right now. Her name is Anna Karvinen, and she works on the Lucas study in Finland. And she's seeing some interesting relationships between a farm-associated microbe that I won't name and protective effects on asthma and WHEEZE. And she actually sees stronger protective effects for a particular oligotype of that microbe versus the others. So it can be kind of a neat next step to take your 16S data to. Okay, and so here's just a LEFSA output. There are different options. You can show a cladogram, which is actually what Don had on his slide, or this other type of output that shows the discriminating features. So this is an analysis from our VDART study, looking at infants that got an LRI yes, versus those that did not. And these are gut microbial taxa that discriminate the infants who ended up getting a lower respiratory tract infection, like Proteobacteria, Escherichia here, and those that discriminated infants that did not experience a lower respiratory tract infection. So again, this is just a type of analysis we could do for the microbiome of the built environment as well in relation to phenotypes. Okay, so here's my last slide that has more questions on it than answers, okay? All right, so once we understand these relationships better, there could be opportunities for intervention. Health disparities research is another important area to think about within these relationships. I showed you that kind of interesting association we saw for race and endotoxin in homes. Compartments, this is really tricky, and more studies need to be done here, particularly looking at the microbiome of the built environment and how it influences the human microbiome. And then looking directly at the microbiome of the built environment and how it influences human health, we have to consider exposure routes. So I mentioned that measurement error correction model and assuming that for the airway irritant effects, it's inhalation exposure. But really for the allergen sensitization outcomes, it could be both inhalation and ingestion. You know, it's hard to know. So timing of exposure matters as well. Specific taxa, picking out the specific microbes that have adverse or protective effects will be important, but it's possible that there are really communities of microbes, mixtures of microbes that are important, not just specific ones. And then the context of the exposure matters too. So the protective communities that exist in homes may be different from those that are present in the farming environment. So that's about it. I have some acknowledgments here. And I know people are probably tired, but I'll take a question or two. Yeah.